This is a conceptual introduction to sex determination mechanisms. It won't be quite accurate in lots of the details, but overall, it'll represent what's going on with genetically based reproductive sex determination in comparison to temperature-based reproductive sex determination that occurs in a lot of reptiles. So genetically based, lots of species, including mammals. Temperature-based, there are quite a few reptiles who use it. For genetic-based sex determination, there are transcription factors that are being produced in particular regions of an embryo that will become, for example, the eyes, the mouth, limbs. And in the region of cells that will become gonads, there's a subset that we'll just call transcription factors A and B. There are also epigenetic modifications and other things that distinguish these cells from others. And of course, cells in the areas that will become eyes would be expressing PAX6, that induces eye development. Limbs would be distalless. So these gonadal cells then are expressing a subset of transcription factors, and it'll trigger genes that require those transcription factors. In mammals, there are 2 X chromosomes in females, and males have 1 X and 1 much smaller Y chromosome. If we zoom in on the Y chromosome, somewhere on it would be a gene, the SRY gene, also called testis determining factor. That's what that name originally was before we identified the gene. And of course, it has a coding and a regulatory region. So what determines whether a particular gene will be expressed, among the important factors, are the transcription factors necessary as enhancers present. And so if TFA in my conceptual introduction and TFB are present in these cells, then RNA polymerase can bind, we'll get messenger RNA, and SRY protein. So in a male, we'll get SRY protein because that gene is here. In a female with only 2 X chromosomes, transcription factors A and B will be present, but there's no SRY gene. So in a female, although those transcription factors would drive expression potentially of genes that are on the X chromosome related to female development. Okay, so what about the next step? SRY protein is a transcription factor. So SRY will bind to parts of genes that induce testis or male gonad structures or other male structures to develop, like the exterior organs related to sex. And so SRY then protein, it's a transcription factor binding to enhancer sites. We'll get messenger RNA and proteins that induce testis structures and male structures. SRY would also be an inhibitor, potentially, for genes for female-specific characteristics. So to run through this, males have a Y chromosome, females do not. So females will have no SRY, whereas males will develop SRY. That's happening only in the region of the cells that will become parts of the gonads, because they have the right combination of transcription factors and maybe epigenetic factors that allow SRY to be expressed. These other cells in other places don't have that same combination, and so only cells in the region where the gonad is going to be will get SRY. SRY, once expressed, then is a transcription factor that causes expression of genes that induce testis structures and male structures. And so we get proteins that are going to cause these cells to develop into a testis and begin the events that will produce male structures here and release hormones and so on to develop male structures elsewhere. And SRY would inhibit genes for female-specific characteristics. In the absence of SRY, then genes on the X chromosomes can be expressed that would be causing female characteristics. So male, SRY. The absence of SRY, and it's not quite that simple. There are some other genes that are important for development of ovaries and female structures, but in the absence of SRY, those can continue, and you can get female structures and a reproductive female. Okay, what about temperature-dependent sex determination? This is not fully worked out yet, so I'll give a conceptual overview. The temperature sensing mechanism, best I can find out right now in 2022, is that these are dependent on temperature-sensitive channels. TRP is the protein family for these channels. It stands for transient receptor potential, which isn't very informative. If this is out and this is in, these are non-selective cation channels. In other words, they allow in often both sodium, calcium, even magnesium. So clearly some negative charges along the channel, but the spacing is such that it's not only sodium, calcium, or magnesium. They are variable, but one category of the TRPs, one group of TRP channels are known to be temperature sensitive. In other words, the rate of random molecular collisions can open or close them. And there's even evidence that single amino acid changes can change the temperature at which these open and close. So you might have, for example, open at greater than 25 degrees centigrade, closed, and closed when less than 25 degrees centigrade. And the signaling, either from sodium entry, calcium entry, probably one of those two or both, that then can trigger the different sexes to develop. So again, we're going to have a part of our developing embryo that will have cells in the region that will develop into gonads. And so these cells will be expressing those particular channels. So if we zoomed in on one of those cells, this cell will have the transcription factors TFs A and B here that will result in expression of the right TRP channel. And so we'll wind up with the TRP channel of that type here. That channel is sensitive to collisions. So at a particular temperature, we'll just focus on the calcium entry. That calcium might end up activating a kinase that phosphorylates an otherwise inactive transcription factor and that could result in expression of proteins for male or female-like behaviors. So this pathway using calcium as a second messenger to do something that might activate a transcription factor, or we could have cyclic AMP involved in this or some other second messenger. This is one of the hypotheses that was out there for temperature-dependent sex determination in reptiles and possibly in other species. Right now, the evidence that I'm seeing suggests that an important part of this, that the calcium and or something related to this calcium signaling is an important part of it, but that it's not actually working in this way. What it's doing is causing an unwinding of DNA. It's an epigenetic activity that exposes a gene for either male or female, a protein that induces either male or female structures. And so in the absence of this pathway, you fail to activate a protein that uncoils the DNA from histones. And so without it, this gene is hidden from view and is never expressed. But when this pathway is activated, that activates a protein that unwinds and disassociates the DNA from the histones. The gene is exposed, expressed, and now you have a protein that will start producing either a testis or an ovary.